Hi, everyone. Welcome to the IEEE SusTech 2021 conference. Uh, uh, and welcome to my presentation. My name is Haoyu. I'm a fourth year PhD student in University of California, Merced. And my major is computer science. Today, my presentation is a reliable tree-level evapotranspiration estimation method for the pomegranate trees by using Lysimeter and UAV multispectral imagery. Before I start, I'd like to say thank you to my partner, Tiebiao, and Dr. Wang, and my PhD advisor, Professor Chen. So in the past few years, the unmanned aerial vehicles, also called drones, have been widely used in precision agriculture applications. For example, the water stress estimation, the pest monitoring, and crop yield estimation, mainly because of the development of UAV technology and remote sensing sensors. The accurate estimation and mapping of ET are essential for crop water management. However, little ET estimation has been studied based on the single tree level or individual tree level. Thus, the purpose of this study was to develop a reliable tree-level ET estimation method using UAV high-resolution multispectral images. So here is my outline. First, I'm going to introduce the ET estimation background. And the second, I'll show you my materials and method, and then the results and discussion. And at the end, I will draw my conclusion and the contribution of this paper. So first, the ET estimation. So ET is known as the main outgoing water flux from the surface on the Earth. And ET is a combination of two separate processes, the evaporation and the transpiration. So mapping the ET temporally and spatially can be useful for many agriculture applications. For example, the soil moisture monitoring, assessing crop water stress, and weather forecast. And there many researchers have been working on this issue, the ET estimation with small UAVs. In 2020, we published a review paper about the ET estimation research with the small UAVs. So there are many different UAV platforms and different sensors for different applications. So if you're interested in this paper, please refer to our uh, review paper. So second, materials and method. So the study set, the flight mission design was tested in a pomegranate field at USDA during the growing season in 2019. So the San Joaquin Valley has a Mediterranean climate with hot and dry summers, and rainfall is insignificant, and irrigation is the only source of water for pomegranate growth. For the pomegranate field, it was randomly designed into 16 eco-blocks with 4 replications to test 4 different irrigation levels. And for the irrigation levels, there are 4 different uh, levels, the 35% of ET, 50%, 75%, and 100%, which was measured by the weighing lysimeter in the field. So for the pomegranate field, there were 5 sampling trays in each block and 80 sampling trays in total and marked with the red labels. And for the drone platform, we used the hover and we built the hover in our lab by ourselves. The hover was equipped with a Pixhawk flight controller, GPS, telemetry antennas, and is able to fly over the pomegranate field for 30-minute flight mission with cameras mounted on the hover drone. And for the multispectral image, we used a red edge M to collect the multispectral data. So the camera has 5 different bands, which are blue, green, red, near infrared, and red edge. It has a spectral resolution of 1280 by 960 pixel with a 46-degree field of view. And also the sensor has a downwelling light sensor, which is a 5-band light sensor that connects to the camera. And the red edge M can measure the ambient light during a flight mission for each of the 5 bands. Then it can record the light information in the metadata of the images captured by the camera. And after the camera calibration, the information detected by the DLS can be used to correct lighting changes during a flight, such as changes in cloud cover during a UAV flight. So for the method, we used the crop coefficient method. So ET estimation by crop coefficient is one of the most commonly used methods for water irrigation management. The crop ET is determined by the crop coefficient, whereby the effect of the various weather conditions are incorporated into the reference ET, and the crop characteristics into the crop coefficient. And as I mentioned earlier, uh, the actual ET for the for our ground truth is measured by the weighing lysimeters. And if we zoom in a little bit, you can see the lysimeter in the field. There are two trays in the lysimeter. And by the way, a lysimeter is a measuring device which can be used to measure the amount of actual ET, which is released by the plants. And by recording the amount of precipitation that an area receives and the amount lost through the soil, the amount of water lost to ET can be calculated. And here is one example for the lysimeter, and you can see it can give us very detailed information about a daily ET. And the third, the results and the discussion. So first, we need to determine the individual tree crop coefficient from the NDVI. So the mean NDVI values for the lysimeter tree are calculated by the near infrared and red band. NDVI is a standardized method to measure healthy vegetation. So when the NDVI is high, it indicates the vegetation has a higher level of photosynthesis. So the figure shows the relationship between the daily crop coefficient and the derived mean NDVI for the tree in the lysimeter with 100% irrigation treatment. The sampling date started on May 8th and ended on October 29th. The linear relationship has an intercept of around 0.61 and a slope of 1.6. It also has a high correlation coefficient. You can see that the R square is as high as around 0.88 or 89. So it indicates a significant correlation between the crop coefficient and NDVI at the individual tree level during the growing season in 2019. So, inspired by the strong linear correlation between the crop coefficient and the NDVI for the individual lysimeter tree, and we calculated the crop coefficient for all the trees in the experimental field by using the linear regression model. So the mapping of crop coefficient and the actual ET were generated respectively, and the value of crop coefficient are mostly between 0.57 and 1.03 on May 8th. So this is an example for the date of May 8th. So based on the crop coefficient, and we also have the reference ET from the nearby weather station, and then we calculated the actual ET for each individual tree in the experimental field. So from the mapping, you can see that the actual ET for individual trees are ranges from 3.2 to 6 millimeter per day. So also by using this is the actual ET mapping for the whole field. And since we have the GPS information and we can get the information for each individual tree. And as mentioned earlier, there are 80 sampling trees in the pomegranate field. 20 of them are irrigated with 100% of ET irrigation. So same with the lysimeter trees. So to validate the linear regression model on individual tree level ET estimation, we also compared the 20 sampling trees with the lysimeter daily ET for the UAV flight days. And the trends of the daily ET for the 20 sampling trays and the Lysimeter tray are shown in this figure. And for the series, each series means an individual tray in the field. You can see the 20 sampling trays have the same trend with the Lysimeter trays. And we also draw the box plot of the Lysimeter trays and the 20 sampling trays. And by the data analysis, we got the R square equals around 0.9, and the mean absolute error is around 0.39 millimeter per day. So which showed a state of art performance to predict the ET for individual tree. So the fourth section is the conclusion and contribution. So in this study, UAV flight missions were conducted to collect the high-resolution multispectral imagery in a pomegranate orchard at USDA. And the experimental results showed that the estimated daily ETC has an R-square and mean absolute error of 0.9 and 0.39 millimeter per day for 100% irrigated sampling trays, which showed a state-of-art performance. And for our future research, we have 3 years data from 2017 to 2020. And we only used 1 year at 2019. So in the future research, we will add more data and to get more data analysis for the results and to get a more robust model. So they only give me around 12 minutes, so I'll stop here and I would like to answer any questions. Thank you.